My name is Yemi Okunsheye. I'll be your program anchor for the evening. Please permit me to bring on board our President, Dr. Kolade Mosoro, for his welcome address. Thank you.
Thank you very much, Yemi. Good evening. On behalf of our Society Initiative for Information, Arts and Culture Development in Nigeria, IACD, I'm very pleased to welcome our viewers in Nigeria and indeed across the world to this program. Our society promotes arts and culture, including scientific culture. Tonight and for the rest of the week, at this time, we'll be examining groundbreaking achievements of the 12 Nobel Prize winners for this year. Tonight, day one, is designated for medicine. Tomorrow, Tuesday, 1st of December, is designated for chemistry. Wednesday, same time, 2nd of December, is for physics. Thursday, 3rd of December, is for economics. Friday is for literature. And we round it all up on Saturday, 5th of December, to cover peace. Our panelists have been carefully chosen. They are all outstanding academics in their relevant fields. We have challenged them to make their subjects as simple and comprehensible as possible so that they can carry the general public along. We operate on the notion that there's nothing abstruse or so technical that a competent disciple cannot bring to the understanding of the layman. That is our charge to the panelists, and I have no doubt that they will do justice to the assignment and keep us very well informed. I would like to welcome and pay very special tributes to our special guest, His Excellency Carl Michael Brandt, the Swedish ambassador to Nigeria, and His Excellency Knut Elv Lein, the Norwegian ambassador to Nigeria, both of whom will be joining us respectively on Friday during the literature program and on Saturday during the peace program. Also joining us expectedly for the literature program on Friday, December 4th, is Professor Wale Soyinka, 1986 Nobel laureate for literature. We thank them all. We equally like to thank our sponsors, Dawn Commission, ZAD Group, and the booksellers for supporting this program. We're immensely grateful to them. Alfred Nobel's earnings and will have generated the most prestigious prizes in the world. It is a bequipment that recognizes scholarship and the best good for humanity for the year. We gather to celebrate the works of these recipients by discussing their achievements. This is a program that we will do annually as long as new and monumental accomplishments are made and recognized the world over. Here, we will follow science and let science and culture interact. Underlying the celebration is a challenge in line with our society's vision for an enlightened Nigerian populace, an encouragement to our academics and students at large to a future of dreams and hard work. There's really no development anywhere on earth that is beyond our thinking realm. For tonight, we will celebrate success and discuss the achievements of the recipients and their relevance to mankind. Again, we welcome you all to a good evening and to a great week. May I now invite that the tape announcing the Medicine Physiology Award winners for 2020 be played. Thank you very much.
You should make me pose. Make it.
So good morning. Very welcome to Nobel Forum for the announcement of this year's Nobel Prize in Physiology or Medicine. So my name is Hans Perlman, and I'm the Secretary General of the Nobel Assembly at Karolinska Institutet, has today decided to award the 2020 Nobel Prize in Physiology or Medicine jointly to Harvey J. Alter, Michael Horton, and Charles M. Rice for the discovery of hepatitis C virus. So here are the three laureates. Harvey Alter was born in 1935 in New York. He performed his prize-winning studies at the National Institutes of Health, Department of Transfusion Medicine, at the Clinical Center in Bethesda, where he remains active. Michael Horton was born in the United Kingdom and performed his prize-winning studies at Chiron Corporation in Embry-Riddle in California. In 2010, he relocated to the University of Alberta in Canada, where he remains active. Charles Rice was born in 1952 in Sacramento in California. He performed his prize-winning studies at the Washington University in St. Louis. In 2001, he relocated to Rockefeller University in New York, where he remains active. Now, Professor Gunilla Carlsson-Hedestam, member of the Nobel Committee, will describe the discovery. So please, Gunilla.
Year's Nobel Prize in Physiology or Medicine awards the discovery of the hepatitis C virus, a major cause of liver inflammation, also known as hepatitis. In their latest report, the World Health Organization estimated over 70 million cases of hepatitis C globally. This is likely an underestimation, as many cases go undiagnosed. Hepatitis C causes 400,000 deaths annually and is one of the most common causes of liver cancer and liver transplantation. There are three main types of viruses that infect the liver: hepatitis A, B, and C viruses. These three viruses are different from each other and belong to different families, and they cause different types of disease. Hepatitis A is transmitted via water or contaminated food and causes an acute infection that typically resolves within a couple of weeks and leads to lifelong immunity. In contrast, hepatitis B and C causes a chronic infection and is transmitted via the blood. Chronic inflammation of the liver is a serious medical condition since the liver is responsible for several key physiological processes. Defects in the function of the liver can therefore be life-threatening. Chronic hepatitis is a silent but progressive disease that destroys the function of the liver over years to decades. Usually without symptoms, but eventually it leads to a chronic, serious condition that can result in cirrhosis and liver cancer. Once the disease has progressed this far, it can only be treated with costly and resource-intensive liver transplantation. An increasing number of chronic hepatitis cases was first noted some 70 years ago in patients that had received multiple blood transfusions. It was also noted occasionally in patients that had received various forms of therapeutics that were purified from donated blood. In 1967, Baruch Blomberg discovered the hepatitis B virus, and he was awarded the Nobel Prize in Physiology or Medicine for this discovery in 1976. This was a key discovery, but it did not explain all cases of chronic hepatitis. The situation was becoming alarming. Because the disease was silent and progressive, it was impossible to know who of all apparently healthy blood donors were carriers of the disease. An improved understanding of blood-transmitted hepatitis was urgently needed to identify blood donors who were carriers. In the late 1960s, Harvey Alter, a clinician who had collaborated with Blomberg, was now working at the NIH Blood Bank. He embarked on an ambitious project to investigate the source of post-transfusion hepatitis. He realized that even after hepatitis B contaminated blood was excluded through testing, most hepatitis cases remained. To determine the magnitude of this problem, he systematically tested the blood supply for the presence of known candidate viruses, and he tracked individuals who developed hepatitis after a blood transfusion. Through this work, he could show that an astonishingly high number of hepatitis cases could not be explained by the known viruses. At this point, he strongly suspected that there was another infectious agent causing the disease. Alter was not alone in suspecting that a mysterious hepatitis cases were caused by an infection, but this was not yet formally demonstrated. In 1978, Alter demonstrated that plasma from patients with this form of hepatitis could transfer the disease to chimpanzees. The animals developed signs of liver inflammation, and this result strongly supported that the causative agent was infectious. Further work by Alter and colleagues over the following years showed that the agent had the properties of the virus. The quest to isolate the virus could now start, but this proved to be extremely challenging and took several more years. The breakthrough came in 1989 when Michael Horton and colleagues working at Chiron Corporation used a combination of molecular biology and immunology-based techniques to clone the virus. That this would work was not obvious, as it depended on two assumptions. First, that genetic material would be present in a collection of DNA sequences isolated from hepatitis-infected animal. And second, that serum from a human with this form of hepatitis contained antibodies that were specific for the virus and that could be used as a molecular hook in the screen. Starting with the blood from an infected animal, the team introduced DNA fragments into bacteria, and where each bacteria expressed an individual protein fragment. By screening with the antibodies, they could isolate one positive clone. This was a high-risk project, but the positive clone turned out to encode for a sequence that resembled sequences of the virus family called flaviviruses. They named the virus the hepatitis C virus. This was the first time these types of molecular-based approaches were used to identify a novel virus. The Horton group immediately took advantage of this knowledge and started to work on establishing a blood test to identify hepatitis C-infected blood. This was urgently needed, and once they had developed the test, they evaluated it blindly on Alter's serum collection from the NIH Blood Bank. This successfully identified all the samples that were suspected to be contaminated, but none of the control samples. The availability of a test that showed this kind of high specificity and sensitivity was a major step forward. Soon, blood supplies around the world could be screened, and this immediately led to a dramatic decrease in the number of post-transfusion cases of hepatitis. One important question remained. Was the hepatitis C virus alone the cause of this form of hepatitis? That is, could a molecularly cloned virus reproduce the disease? Charles Rice, then working at Washington University in St. Louis, and others investigated the molecular requirements for viral replication. They identified a highly conserved 3' end of the viral genome, which they thought was important. When Rice and his team tested these clones containing this region, they first could not observe replication in animals. Rice then speculated that the clones they had used contained inactivated mutations somewhere in the genome, shown here as red dots. He knew that RNA viruses of this type are error-prone and acquire random mutation in their genomes during their replication. Production of such genetically diverse swarms of viruses benefits the virus population as a whole, but it means that individual clones can be defective. He therefore sequenced many clones and compared them to each other. He observed that several of these clones contained potentially inactivating mutations, which he could remove through genetic engineering. When he combined such a repaired genome with a 3' end of the genome, he had created what he hoped would be a functional virus. When this genome was injected into the liver of chimpanzees, he observed clinical signs of hepatitis. An infectious virus was now present in the blood, and it persisted over time. This provided conclusive evidence that the cloned hepatitis C alone could cause the disease and induce a productive infection. The laureate's achievements provided the foundation that was needed to now start combating the spread of the virus. Thanks to the effective blood screening programs, the hepatitis C virus is now almost eliminated in many parts of the world, and the development of highly effective antiviral drugs means that more than 95% of treated patients can be cured from the infection. These developments have saved millions of lives worldwide. Continued efforts to implement blood screening programs and treatments globally raise hopes that hepatitis C virus can be controlled and eventually eliminated. To summarize, the pioneering work of this year's laureates is a landmark achievement in our ongoing battle against virus infections. Starting with Harvey Alter's demonstration that a new virus was responsible for a majority of post-transfusion hepatitis cases, and Michael Horton's groundbreaking work to clone the virus and develop a blood test that could be used to exclude contaminated blood. And then leading to the elegant work by Charles Rice, showing that a molecularly cloned hepatitis virus alone could cause persistent infection and reproduce the disease observed in humans.
Once again, good evening, ladies and gentlemen. Please permit me to introduce our panelists for tonight, Professor Jesse Otegbayo, the CMD of UCH, for his presentation, and to bring on board our moderator for tonight, Professor A.A. Ismaila. Thank you. Welcome, gentlemen.
Thank you.
Thank you.
You are on, sir. Professor, you're on, sir.
Is it the moderator now, I guess?
I thought we should just go ahead and have the lecture by Professor Otegbio.
Okay. All right. Good evening, ladies and gentlemen. I would like to appreciate the organizers of this program for inviting me to be a part of this laudable program. I have some slides to share so that I will speak to it as I speak on. I mean, as I project it and change the slides. I'm trying to get my slide on the screen. Can the host help me to share my slide? Hello?
Is the host hearing you so that they can make you a co-host or they share it for you?
We already made him co-host.
Okay, so share your slide or take by.
Okay. A little hitch here. Share screen. Microsoft. Sorry, I'm Google Dropbox. Wow. So sorry, I have the slide on my screen. I'm trying to get it on.
It's on now.
Sorry?
It's on the screen now.
You can open your presentation now.
Yeah, that's what I'm trying to do.
Is it the one at the bottom already that is minimized?
No,
okay,
it's opening now. Are you seeing it now?
Yeah, we can see you. We can see your screen.
Yes, sir, we can.
Okay. Well, I'm to speak on the Nobel Prize for Medicine or Physiology for the year 2020, which was awarded to Harvey Alter, Michael Houghton, and Charles Rice for their discovery of hepatitis C virus. I would like to speak a little about what the virus is all about because we have a mixed audience, and also to mention the organ that the virus particularly affects. And the major organ, even though it can affect other viruses, other organs in the body, but the major organ that the virus affects is the liver. And that is why the infection or the disease it causes is called hepatitis. And hepatitis is from two Greek words, hepa meaning liver, and itis in medicine is referred to inflammation or refers to inflammation. The liver is the largest solid organ in the human body, and it is located in the upper part of the abdomen or stomach on the right side, protected by the diaphragm and the ribs. This is because it is an essential organ. An essential organ is an organ that the body cannot do without, or the human organism cannot survive without. There are some organs you can remove from the body, and the human organism still continues to live. For instance, you can remove the entire spleen in the human body, and it has no serious consequence. But without liver, there can be no life, and that is why it is well protected. It has many functions, of which I will just mention a few here. The liver removes potentially toxic byproducts of certain medications or metabolites. It also breaks down nutrients. When you eat, the food consists of mainly carbohydrate, protein, fat, minerals, and vitamins. Many times, these nutrients cannot be used or utilized by the human cells until it is processed by the liver. And that is why the liver is often referred to as the energy generating set or the powerhouse of the body, because this is why the units or the currency of energy is generated. It also helps our body to fight infections by filtering bacteria and other organisms, including viruses and fungi. So it has immunological properties, and it produces most of the substances that regulate blood clotting. I will not bore you with the details, but that is just on the surface. And it also produces a substance called bile. This is the greenish substance that is found in the gallbladder, and it is a waste product of breakdown of blood cells. And of course, it's also involved in the absorption of fat and some vitamins that are essential to life that are called fat-soluble vitamins like vitamin A, D, E, and K. All the proteins that the body uses as a building block to build our tissues, our cells, our muscles, are also produced in the liver. Of course, it also performs some storage functions for the time of need. It's like a bank also in the body. So it has various functions that are essential to life and living. I've mentioned a few things about what is on this slide. And there are various hepatitis viruses. In medical parlance, we categorize them into two. We call them hepatotropic viruses and non-hepatotropic viruses. Hepatotropic viruses, as you may guess from the word hepa, are the viruses that thrive mainly in the liver, while non-hepatotropic viruses thrive in other organs mainly, but may affect the liver. And mind you, I also said earlier that these hepatotropic viruses, of which hepatitis C is one, also affect other organs. But it's like the viruses called hepatotropic viruses bite the liver and leak other organs, while the non-hepatotropic viruses bite other organs and leak the liver. That is, the severity of these viruses is more on the liver compared to the non-hepatotropic viruses. Basically, we have 5 of these hepatotropic viruses that survive mainly and multiply in the liver. They are named after the alphabets in the order in which they were discovered. So there are hepatitis A, hepatitis B, hepatitis C, hepatitis D, and hepatitis E. But of course, there are other viruses that also affect the liver that are not hepatotropic viruses, like I mentioned, like herpes virus, Marburg virus, and so on. So the A virus is transmitted through water or contaminated water and food or stool. And that is why it is said that transmission is by fecal oral route. That is through the feces that is passed that contaminates either vegetables, water, or any kind of food, or through touch of that organ and the handling of what is edible. But it can be prevented with good hygiene, and it has effective vaccine. Hepatitis E is similar to A in terms of their nature and activity in the human body, and it is also transmitted through fecal-oral route. The others, like hepatitis B, hepatitis C, and D, are often called serum hepatitis because they are only transmitted through blood and blood products. For hepatitis B, many people get infected before the vaccine was widely available. In developed countries where vaccination of newborns is mandatory for the various infectious agents that are well known, the infection is almost completely eradicated with a very, very low level in their society. Like in the United States, the United Kingdom, the incidence of hepatitis B is less than 1%. But in developing countries, Nigeria inclusive, because of poor utilization of available vaccines and sometimes vaccine failures, quite a high number of our population has hepatitis B. For instance, in the survey by the Federal Ministry of Health in 2017, the prevalence of hepatitis B in our community is 11%, which is quite high. And over 80% of people who are affected are not even aware. And that is why it is important for us as adults, one, to ensure that our children, when they are born, are vaccinated appropriately. It takes about 3 vaccinations spaced in different months. And of course, those of us who are adults who are not sure whether we had hepatitis B vaccination or not, we have to screen ourselves. The screening methods are now very much available. We have it at the University College Hospital and other self-respecting laboratories. And vaccine is also widely available and is over 90% efficient. For hepatitis C, which is the major topic of today, thanks for the discovery of the screening methods by Alter, Putin, and Rice, we can now screen for the hepatitis virus, and this is also available. But the unfortunate thing that there's no vaccine available for this type of virus, because hepatitis C is not just a single virus. It exists with multiple viruses, quote unquote, or I would say it's a kind of quasi-species virus in which you have different types of genomic constitution of the same virus. So if you provide or produce a vaccine for one particular one, it will not capture the other. And that is why there has been no effective vaccine for hepatitis C. However, research work still going on to ensure that the vaccine for hepatitis C see the light of day. So in summary, what I've said, hepatitis A transmitted through fecal-oral route, most of the time it is self-limiting. However, it can be severe in those who are immunosuppressed and also sometimes in pregnant women. For hepatitis B, it is mainly transmitted through blood and occasionally through sexual intercourse. What is called vertical transmission is transmission from mother to child during the process of birthing, and it's the leading cause of primary liver cell carcinoma or hepatocellular carcinoma in developing countries. Over 80% of our patients with liver cancer are also positive for hepatitis B. And of course, it also causes liver cirrhosis, which is scarring of the liver. The liver is a fleshy or meaty substance. It's a delicacy actually because a lot of people love to eat it. It's also rich in vitamins and all those substances I mentioned. For hepatitis C, it's also transmitted in the similar way to hepatitis B, except that it is rarely transmitted through sex. And the incidence is higher in developed countries than in developing countries because it is mainly transmitted in those populations through intravenous drug injections. Graciously, intravenous drug abuse is not as common in our climate compared to developed countries. I think this is because these drugs are quite expensive. It's only those who are blessed financially who can engage in it. In developed countries, it's the major cause of liver cancer and cirrhosis. And it's a reversal of what happens in our own environment in developing countries. The Ministry of Health publication I mentioned earlier on got a percentage of 2% of our population having hepatitis C. Then the third one, which is hepatitis D, also called delta hepatitis, is a freak in that it cannot live on its own. It has to latch onto hepatitis B, either by super infection, that is, in an existing hepatitis B individual, hepatitis D now super infects, or co-infection, in which case the virus, that is hepatitis D, is transmitted together with hepatitis B ab initio from one source to another human source. And it is also a serum hepatitis that is transmitted through blood or blood products. I've mentioned earlier on that hepatitis E is like just like hepatitis A, except that the severity is usually higher and the mortality also so in pregnant women. And it's much more common in the Indian subcontinent in Southeast Asia. Now, for what we are talking about Nobel Prize in Physiology of Medicine, the prize was first awarded in 1901, and it was awarded to Emil Adolf von Behring because of the work he did on serum therapy. You know, if you hear that some people are experimenting with some form of therapy for this COVID-19, that is, you take the serum, that is a filtrate from the blood of someone that has been infected and use it to treat others. It is believed that because of the antibodies that that individual has developed, it can cure the virus. We learned that this therapy was given to President Trump when he became positive for COVID-19. And the award has been given in 111 times to 222 Nobel laureates between 1901 and 2020. The latest one, which we are talking about today, was won for the discovery of hepatitis C by this trio of Alter, Hutton, and Rice. Just a little about the background of the Nobel laureates for 2020. I will not mention or say everything that is on the slide, but you have the slide and you can read it later. I just want to emphasize something which I see as not being practiced in our own environment, and which has, in a way, whittled down our ability to make progress in science and research. If you look at it, Harvey Alter was a physician, or is a physician, a hematologist that is a specialist in diseases of the blood, whereas Michael Hutton and Charles Rice are both biochemists, and Rice on his own is also a zoologist. And they worked together and in sequence, not in parallel, to discover at the end of the day hepatitis C, which is a major breakthrough that led to the identification of drugs, that is drug development, that has made it possible for hepatitis C to be a curable disease. Before this happened, millions of people have died, and as at date, over 70 million people are still infected with hepatitis C. Now, what did they do? The work of Alter, Hutton, and Rice characterized the form of hepatitis C to be a distinct clinical entity caused by an RNA virus. RNA is ribonucleic acid virus of the family of viruses called flavivirus or flaviviridae. And yellow fever virus also belongs to this group. Actually, one of the researchers was working on yellow fever when he serendipitously discovered that there was another virus, that is Hutton, and this led to cloning of that virus through molecular and immunologic methods. So the work paved the way for the development of screening methods, which also paved the way for development, sorry, that's an error. The screening methods have reduced the risk of acquiring hepatitis from contaminated blood. Let me say something a little about contaminated blood. Blood, as of today, cannot be manufactured. It cannot be produced. You can only get blood from another human being that is carrying blood in its veins. That is one thing science has not been able to break through. There are substances that have been developed to look like blood, but they never perform the same function or have the same effect or have the same constituent as blood, because there are thousands of substances and cells in the blood, which is something very difficult to put together by science. And blood is equivalent to life. We often say that something is the blood life of something. It's like without blood, there's also no life. Just as without liver, there can be no life. And their main or major work, that is this scientist, was on post-transfusion hepatitis, because they discovered that when they take blood from someone and give to another person without being screened for these viral agents, the individuals develop yellowness of the eyes called jaundice. They develop fever, loss of appetite, nausea, vomiting, stooling, abdominal pains, and all the things that accompany the disease entity called hepatitis, including weakness and so on. Then they felt that there must be something in this blood that is causing this yellowness of the eyes. And that's one thing about science. Inquisitiveness and deductive reasoning makes one to develop, and also for one to have a passion to look for solutions to certain problems. And that was what these people did, that they were able at the end of the day discover what has earned them this Nobel Prize. And I would say that this has also led to the development of effective antiviral drugs that have improved the lives of millions of people across the globe. I will stop there, but before I stop, I will just say that, like I said earlier on, they worked in sequence. Alta was working on lipids, on serum of post-transfusion patients who developed hepatitis, and he discovered that there are some lipids lipids are like fat in the blood, and he discovered that there are some particular lipids which he knew were usually found in viruses. So he felt there must be a virus in this sample, and this led Hutton to take it off from there to use molecular methods, which led him to discover that there is an RNA, that is ribonucleic acid virus, of which hepatitis is one in the serum sample. So the third person who took the baton, that is Rice, now used genetic engineering, sequencing to define the particular virus. They were able to say that, and together they came together, they were able to work on these findings to come to the conclusion that there's an entity or a new virus which is different from the two viruses that were known as at that time. As at that time, naming them in alphabetical orders, the two known viruses were hepatitis A and hepatitis B. And initially they called it when they had not defined the hepatitis completely, they called it non-A, non-B hepatitis virus. That is, it is not hepatitis A that we know. It is not hepatitis B that we know. It is non-A, non-B. But subsequently, they were able to define that this is a viral entity separate and different from the initial two. And at the end of the day, it was named hepatitis C. It was later that hepatitis D was discovered in Italy, and hepatitis E also followed. However, in spite of all this, there are still some viruses that are yet to be discovered, because about 10% of post-transfusion hepatitis, that is hepatitis that occurs after blood has been given, it's been found that one may not even be able to detect any of these serum viruses in the blood of those individuals, meaning that there are still some things some viruses that are still yet to be uncovered. There have been postulations about certain entities of viruses, but science has not agreed that they are real virus on their own. And like I said, research still going on. But just to mention also that there are some agents that cause hepatitis that are not infective in nature. That is, they are not due to infective agents, either viruses, bacteria, fungi, protozoa, and all that. One of them is alcohol. Alcohol can cause hepatitis, and it can also lead to cirrhosis, and can also lead to liver cancer. Drugs, there are certain drugs, including paracetamol that we take for granted, can cause liver disease, can cause hepatitis, can cause chronic hepatitis. We can progress to advanced liver disease. And of course, there are so many other things. In order not to take your time, I will stop here and probably take questions and answers. Thank you.
Thank you very much, Professor J. Otegbayo, for this excellent summary of the Nobel Prize winners for medicine or physiology for 2020 that was awarded to these three gentlemen or scientists of international repute that have brought relief to the world decades after a lot of concern were raised about post-transfusion hepatitis. I've seen only one question in the chat box so far, so I will start with that one before I ask some questions on behalf of the audience. The first question here is hinged on your last or concluding statement where you say that about 10% of post-transfusion hepatitis are still not linked to identifiable viruses, including the C that we've just talked about. And therefore, Kolade Mosuno sent in a question, the only participant that sent in a question so far. And Kolade is asking that with the nomenclature of hepatotropic viruses, named A, B, C, D, and E in that order, does that mean or does it suggest, with also your 10% that have not yet been discovered, could we be having A to Z hepatitis lurking that is unknown on the horizon? So maybe you start with that before we go to other questions that are jotted down on behalf of participants, Mr. Dr. Otegbayo.
Well, I don't have the power of prophetic pronouncement. It's difficult to define what science may reveal to us in the nearest future or many years to come. Because today we have access to polymerase chain reactions using molecular methods to diagnose. And we are limited by the capacity of this kind of test. We cannot tell. In another 10, 20 years, someone may discover, and I'm sure research work is going on in other climes, and someone may come up with another method of detecting these viruses that will be more sensitive and more reliable than what we have as of today. And with that tool, with that scientific tool or molecular tool, as the case may be, they may be able to discover more viruses. So like this year now, I'm sorry, last year, SARS-CoV-2, it just came from the blues. Even though coronavirus had existed before, then suddenly, either by mutation or otherwise, a new virus just came on board and it dribbled the whole world, and we are still battling with it. So I cannot tell, but it's not impossible that A to Z may occur.
Maybe in the future we'll see more of Croton, Alta, and Rice coming up with some other viruses that will be named in that order respectively. The next question I will ask on behalf of participants. We know that hepatitis C is relatively low in Nigeria in terms of prevalence. You talked about about 2%, unlike hepatitis B, which is about 11%. But there are pockets of regions in Africa, particularly Egypt and part of Cameroon, that have particularly troublesome high prevalence of hepatitis C. It will be good, Professor Otegbayo, to tell the audience why those regions have relatively high HCV compared to the rest of Africa.
Yeah. Thank you very much, Professor Samala. In Egypt in particular, they live close to the River Nile, and there's a disease called schistosomiasis, which is a parasitic infection that can enter through the skin, usually the feet or any part of the body that is submerged in infected water. And when they go into this water, they get infected with schistosomiasis. And in the times of old, though the practice is changing now, what they used to treat is such that they use a parenteral way of treating the infection, which leads to shedding of blood and transfusion of the blood infected with hepatitis C to another person. So this is why it's actually the treatment of this schistosomiasis that has led to the high prevalence of hepatitis C in that population. And of course, things have changed. The prevalence is going down because the government of Egypt saw it as a major health threat. And they have done something about it. They have partnered with drug companies to make sure that the drugs for the treatment of hepatitis is affordable, even by the LIGs, the low-income groups. And in many instances, it's even free. So because of the aggressive nature of the approach to the treatment, the prevalence is going down, even though it's still higher than in the other parts of Africa. Thank you.
Okay, thank you very much. The other question that has just came in, one from Jide Adefope and Muli Ademola, they're asking, even though hepatitis B was discovered much earlier than hepatitis C, why is it that up till today, we can talk about cure for hepatitis C virus, while the cure for hepatitis for B has eluded us? And the same thing, that follow-up question, which you can take the two together. Why is it also that we have vaccine for B, on the other hand, and still no vaccine, which you had addressed in your lecture? But I think it's good to take the two questions together. Why is there no vaccine for C up till now, while there is vaccine for B, which is effective, and the other way in terms of treatment and cure?
Okay, let me take that of hepatitis C, which I think I explained earlier.
Yeah.
Hepatitis C is not just a single virus. It's a virus that has multiple quasi-species, that is coexistence of some genetically different viruses existing together. Therefore, when you provide or produce a vaccine against one of the viruses, does not capture all the viruses. So there are some parts of a virus that contains critical genomic materials for the survival of that organism. And those are the areas that vaccines target. Like, it's like if you remove the heart of a human being, just the heart, the man will be dead. You can cut the thighs, you can cut the legs, remove the eyes, and that individual will still be alive, though the quality of life may be suspect. But for this virus, because of the multiple nature of the virus that coexist as as hepatitis C, it is difficult to produce a single vaccine that can take care of all this. And that is why the search for effective vaccine against hepatitis C is still ongoing. And I'm sure one day we'll hit the bull's eye. For hepatitis B, the reason why it has not been possible to cure, hepatitis B occurs in various organs, although the largest concentration is in the liver. And one of the genomic materials of hepatitis B is called the CCC DNA. That is the covalently closed complementary diribonucleic acid. That's the CCC DNA. This particular aspect of the virus, which is one of the most complex viruses ever known to man, because it has various genetic materials, lives in the core of the liver. And it is, till date, not possible for drugs to reach this CCC DNA in the liver. So when you treat the viruses within the blood or within the serum and other superficial tissues, these ones that are deep in the hepatocytes, that is the liver cells, cannot be reached by drugs. And that's why it is not curable. Hepatitis C does not have this type of genetic material, and therefore it is easier to cure. Even with direct acting antiretroviral drugs these days, in 6 weeks to 3 months, you can achieve 95% cure of hepatitis C, whereas hepatitis B cannot be cured. However, the story doesn't end there. With the drugs we have now, the virus could be suppressed. That is, you prevent the virus, the drugs prevent the virus from multiplying. And therefore, this cccDNA is just like a sanctuary in the liver. The drug does not get there, but the drug prevents it from multiplying. And once a virus cannot multiply, it has lost one of its major weapons. Because even these days in social media, when you say something goes viral, it means it has multiplied tremendously. By the time you disable the virus from multiplying, then you have kept it in check. The problem is just that with available drugs now, we have to take the drug for life, which is not the problem, because there are many other diseases too, like hypertension, diabetes, whereby the treatment is for life because they have no cure. So as of today, hepatitis B has no cure. Just like HIV also, when you start treatment for HIV, which is also a virus, the treatment is also for life. You live a healthy life, the virus does not multiply. In fact, sometimes it's not detectable. Not detectable does not mean the virus is not there. It only means the level is so low that it cannot cause any harm to the individual who is carrying the virus. My take, thank you.
Okay, thank you very much. Jide Adefope still is saying that the current pandemic of COVID-19 or the SARS-CoV-2 that has taken the world with a storm is linked to messenger RNA, which hepatitis C also, which was discovered much earlier, is also an RNA virus. So Jide Adefope is asking, why do we have rapid development of a number of candidate vaccine against coronavirus, but yet not yet against C? So I think you have addressed most of the terms of the cause species and subspecies of C, which may not be that as varied as so complex as that of hepatitis C. But I think the other question that I want you to address is the fact that while hepatitis B can just come up with cancer in an individual without a pre-existing cirrhosis, hepatitis C always have to pass through the pathway of cirrhosis before you have cancer. I think it's very important so that people will know that this virus B can just come up with a cancer without initial warning of cirrhosis. But C does it the other way after decades. Thank you.
Thank you very much, Professor Milan. You are very correct.
Okay, we go ahead. I thought you were going to have a comment for Adefope.
Oh, okay.
About the corona and yes, the
you answered him. They said necessity is the mother of invention. Because of the way COVID-19 infection is decimating the population, the scientific world rose aggressively to find a solution. And for the first time in human history, scientists were able to develop a vaccine that in about 90% can protect against COVID-19 in less than 2 years. Most of the time, the average time for development of vaccines is about 5 years or more. Yeah. But because of the nature of this pandemic caused by COVID-19, scientists rose, and they've succeeded to a very great and large extent. To say it again, probably the third time. Yeah. Viruses, even though they bear the same name, virus, it's just like human beings. If you go to the Wolof people in Senegal, you see some are so dark like charcoal. You go to Egypt, you see they are very fair. We are all human beings, but with different morphology and anatomy. So same goes for viruses, even though they are all viruses, but their genetic constitution, their form, their body form vary. And what is called the critical part of their genome is also different. If, for instance, you have a virus that is shaped is just like a circle, there will be a particular protein that is so important to that virus to survive that if you use a drug to hit that particular one, the virus will not succeed. And that may just constitute about 5% of the entire circle of the virus. If you are using drugs that will take care of the rest 95% without hitting that particular critical protein, you will not succeed in curing the virus. So what I'm saying, hepatitis C is a virus that has multiple quasi species. That is a constellation or a combination of many viruses in one. So which one do you attack? And it has a very complex structure that woman showed. It has a 5' end, and it has a 3' end, and of course, many structural elements in between. And it is difficult to find a vaccine that would target all these proteins at the same time. And that is the complexity that it has given to the vaccine development for hepatitis C in particular. But like I said, we are not to lose hope. Scientists are still at work looking for a particular vaccine that will be able to catch multiple sites in the constitution or the morph of the virus. And yes, when that is achieved, I believe we will find a vaccine.
Yes, I cannot agree with you more because if you look at, apart from the viruses being different in terms of the biology and other properties, the circumstances of C and the impact it has on the world compared to corona, they are totally different. The urgency, the community transmission, the air droplet transmission made the drive to get vaccine for corona more desperate and more urgent than C. But I think we still remain hopeful there are some candidate vaccine in preclinical stages now. I want to ask the last question. Somebody sent in Kolade Mosuno again. He said, we have seen collaboration where you have a clinician who started as a hematologist, collaborated with a basic science scientist in terms of microbiology and genetics. And they came up with what was disturbing the world with a solution to hepatitis C in terms of diagnosis, treatment, and now we are talking about potential vaccine, hopefully in the future. So Kolade Mosuno is asking, how do we encourage such collaboration in Nigeria in particular, and sub-Saharan Africa in general, just as we said earlier, that these are not sort of things that are common in our climate, where you see people across specialties, across major branches of science coming together to attack and answer a particular research question. So that's the question Kolade Mosuno is asking. And I think you can address that. That should be the last question that I have from my end. And there's none in the box now. So this should be the last answer we expect from you, Professor Tekbai.
Well, thank you very much, Professor Samaila. When there is a will, there will always be a way. The first thing is for scientists to know that no man is an island and no single individual is a repository of knowledge. And that there's a need to collaborate and to work together, not just when we have become scientists, but even when we are students. There's in UCH here, we have a students association called USA, University College Hospital Students Association. And I'm always encouraged when I see them because they play football together, they work together. Here you have a physiotherapy student, a medical student, nursing student, lab scientist, forgetting their background in terms of their areas of study. They do everything in common. They have parties and management supports them because I love such a thing. I mean, by the time they have grown up becoming scientists, you will discover that it will not be difficult for them to work together. What I see in our climate is that people want to be champion and they want to work in silos. It's like, I have this knowledge, let me use it to the utmost best for myself. And when people have that mentality of me, myself, and try to do things to acquire fame by themselves alone, it will not work. So I think the way forward is to start grooming students right from the time they are in school, that basic scientists need to work with the clinicians to get to the roots of problems. I mean, as clinicians, I'm a clinician. In the course of my work, I've had to work with so many basic scientists, both within Nigeria and outside. In fact, I wasn't satisfied with my clinical qualification alone, so I went for a master's in a different area. I went for PhD. And today, one of the reasons I was able to attain the status I've attained is working with them, because we're able to publish so many papers together, which mainly are a combination of basic science and clinical science. But I wouldn't say everybody's like that. And the earlier we drop the idea of me and myself alone, the better for us in the scientific or research world. Thank you.
Thank you very much, Professor Otegbayo. I think your response further stressed the common saying that if you want to go fast, you go alone. But if you want to go far, you go together. I think that is the essence of collaboration, that is of working together. I would like to...
Professor Ismail,
thank you.
Professor Ismail, before you close,
Yes.
I think that Professor Femi Bamiro would like to ask a question.
Oh, great. Go ahead. You're welcome, Professor Bemi.
Bamiro.
No,
Bamiro, yeah. Hello, Femi.
Yeah, actually, it's not a question. It's the last issue being discussed, and that is this question of people collaborating in research, multidisciplinary.
Yes.
I mean, it's been a big problem in this, getting our people to work together, but gradually, I mean, because there's hardly any problem that you can solve just from one discipline. And maybe you have heard recently, well, as recent as last week, TED Fund constituted 12 centers of excellence, 2 centers per zone. And in the Southwest, UI was made one of the, given one of the centers of excellence, I think UNILAG. And the area that UI is supposed to work is in the area of multidisciplinary studies. How do you bring people together, to work? In my own experience, when I was Vice Chancellor, was thinking to go by, there was a time, Makato, they gave us a grant to establish Center for Sustainable Development. And in that, and there we're supposed to produce people with masters in development, sustainable development. And they realized that development is about people. It's not about only your economy and so on. So the curriculum was designed in such a way that it was interdisciplinary. And they said that we should, when we were writing the proposal, they said we should bring people from economics, from health studies and engineering and so on, together for the goal. So, suffice it to say that I found that, as you have said, silo. If you don't centralize it, if you give it to one department or so on, the others will not collaborate. So I had the being of, PG school there, to coordinate. And we brought representatives in that proposal writing, representatives from economics, from engineering, from health sciences, medicine, and so on. Suffice it to say that our colleagues from economics, they told us that they will not participate unless that program was located in economics, because development is economic. And we try to make them realize that no, economy is not the only thing. Before, yes, people were equating it to economic, but no longer. You know that they did not participate. That was the extent. So, but suffice it that at the end, we won the proposal, and they were now forced to participate in the system. But another thing that also something that is discouraging multidisciplinary, each university we have to look at its reward system. The reward system, when people, academics are being promoted, we tend to, the system tends to discourage multi-authored papers. When you say, oh, what is your share of contribution? You must divide the 100 and come and see people fighting I mean, you have a paper probably by maybe 4 or 5 authors, and then one will be saying, no, mine is 50%, mine is 60%. I mean, that type of thing. But in other crimes, they have solved such problems. You make sure that at least those who have contributed, it's like they are credited, with the quality, rating on their bet. It's not the same thing here. So, I mean, there are issues, but I hope that you have been given this Center for Multidisciplinary Studies will be able to promote, people working together, because as Professor Tegbaya has said, I mean, most people are still working in silos and you don't go far. And as mentioned. So it's something that we have to look critically. Otherwise, working together is just key to making, progress as already demonstrated by the Nobel laureates. Thank you.
Thank you.
Professor Mala, I have a comment.
Okay, go ahead.
Thank you very much, Professor Femi Bamiro, my former Vice Chancellor. The second part of your comment was what I was going to speak to before. However, you were our Vice Chancellor some years back. And like you said, when it comes to the reward system, that is the promotion, they talk about percentage contribution. Mm-hmm. And when you have a paper where there are multiple authors, sometimes you are given as low as 5% if you are far ahead. And sometimes the person who is the lead author, they say will get maybe 50%. And if you are the sole author, you get 100%. No matter how bad the paper is, once it is accepted for promotion exercise, is 100%. This is very discouraging. And for us in the College of Medicine, there's no way you would not do multidisciplinary studies. It's just not possible unless you are cooking results in your office. If I'm a gastroenterologist and liver disease specialist, there's no way you do some procedures and not have a pathologist in the laboratory who is in basic science to help you look at those tissues, interpret them. You will now marry the clinical picture you are seeing with the histopathological findings in basic sciences. Then you write your paper. And you may even have others in, like I work with people in pathology who look at the tissues we take. Then in virology department, they are not even doctors, medical doctors at all. I have to work with them to see that they have this particular virus. Then I take a sample of the liver, I take it to a colleague who is a pathologist. We all put everything together. At the end of the day, you'll be having multiple authors. But when the reward comes, you are so discouraged. I think with the kind of voice you have at the University of Ibadan, I think you should be among the counted to speak against this suppressive and depressive method of assessing or rewarding people when it comes to multidisciplinary issues. Thank you very much, sir, for appearing in this program. Thank you, sir.
Thank you.
Thank you very much, Professor Tekbayo. I think I would like to conclude my own function by thanking the Initiative for Information, Arts and Culture Development in Nigeria, IACD, for giving us this opportunity. This is a very important platform that I think we need to bring in young scientists, young career academics to be able to know about the existence of this great initiative and this type of program. I can see we are less than 50 as participants. Unless we limited the number, I think as we go into chemistry, physics, economy, literature, and peace, subsequently from 1st to 5th of December, I think the organizers in IACD should ensure that these programs, this Nobel series are popularized among students and young researchers so that we will see hundreds of participants, unless we are limited by space from the host. Thank you very much. Thank you.
Thank you, Professor Ismail. We're very grateful for your advice, and we're very grateful to Professor Otegbayo for the presentation. It's a new journey that we're embarking on, and we're very happy to have you on board. Thank you. We'll take your advice seriously. On behalf of the board and the executive, I want to say a big thank you to everybody, especially to our presenter and our moderator, and to our sponsors too. because we're a non-profit organization. Anything and everything goes a long way for us to be able to achieve our aims, objectives, and desires. And we really thank our participants, everybody that's contributed tonight, that are on board. We're very grateful, and we look forward to seeing you tomorrow again and through to the weekend. Thank you. God bless, and do have a good night. Thank you.
Thank you very much.
Thank you. Bye.
Bye-bye.
Bye, Professor Samaila.
Thank you, sir. Thank you very much, sir. Thank you.
Yeah, nice seeing you.
Thank you,
I've
sir.
heard your
Thank you,
name
sir.
before, but
Thank you, sir. Thank you, sir.
Thank you, Professor Samaila.
Thank you, sir.
And thank you, Professor Tegbayo.
Thank you, sir.
The pleasure is mine, sir.
Yeah.
So
What I always say about people like Otegbayo, may I not need your services?
Well, we also do preventive gastroenterology. So we can counsel you.
Okay. All right. Okay. Maybe the counseling side.
And may he be there should you need his services.
Yeah. Thanks, everyone. Bye-bye.
Bye.
Bye. Thank you. Thank you.
Bye. Well done.